Hi friends, today we are going to see the past year NEET questions for the chapter Molecular Basis of Inheritance, and today in this video we will complete part 1. So let's begin with the first question, which was asked in the year 2017. The final proof for DNA as a genetic material came from the experiments of Dash. So who carried out the experiment and proved that DNA is a genetic material? So what is the full form of DNA? It stands for deoxyribonucleic acid, and that is the genetic material which is inherited from generations to generations, and it's a coding, right? It has genes in it. So this was proved by Hershey-Chase experiment, where a series of experiments were conducted in the year of 1952, and the full name of the two people who conducted this is Alfred Hershey and Martha Chase, that helped confirm that DNA is a genetic material. So you can ask me, what do they do here? So these two people concluded that DNA, not a protein, was a genetic material. They determined that the protective protein coat was formed around the bacteriophage, but that the internal DNA is what conferred its ability to produce a progeny inside a bacterium. Therefore, the right answer is option A. Next question here, again asked in the year 2017. DNA fragments are Dash. So before we go into what they are, so what is DNA? How can you describe DNA? See, DNA is made up of deoxyribose sugar, right? Deoxyribose sugar, and it has a phosphate group and it has a nitrogenous base. Phosphorus groups are negatively charged, which will impart. So this negative charge of the phosphate imparts a negative charge for the DNA. The charge of the DNA and the DNA fragments are the same. Therefore, DNA fragments are negatively charged. Therefore, the right answer is option A. Next question, again asked in the year 2017, sorry. Which of the following RNAs should be most abundant in animal cell? So you have different option here: transfer, messenger, mitochondrial, ribosomal RNA. So which one you think will be the right answer here? Yes, the right answer is option D, rRNA, right? The right answer is rRNA. What is the full form of it? It is ribosomal RNA. So they say that it constitutes about 80% in animal cell. Ribosomal RNA, which is also called as rRNA, is the most abundant in animal cell. It constitutes about 80% of the total RNA in the cell. Therefore, the right answer is option D. Next question. And if you continuously see here, you're asked about 4 questions from this chapter, Molecular Basis of Inheritance, continuously in the year 2017. So when it comes to the NEET aspect, it is good that you concentrate on this lesson, because you might expect some 2 to 3 or sometimes 3 to 4 questions from this chapter. So here, look at this question here. The association of histone H1 with a nucleosome indicates Dash. So you have 4 options here. So what are histones basically? They are proteins, right? So what else you can talk about histones? Any other points that you can remember here? Histones are highly basic proteins, and they are exclusively found in the eukaryotic organisms, right? Eukaryotic organisms, and they help in the package and order the DNA into structural units called as nucleosomes. They are the chief protein components of the chromatin, acting as spools around which DNA winds and playing a role in gene regulation. So I've given you an intro about histone. So the association of the H1 protein with nucleosome indicates that the nucleosome which requires DNA condensation. Therefore, the right answer will be option B. The DNA is condensed into the chromatin fiber. Next question. A complex of ribosomes attached to a single strand of RNA is known as Dash. What do you call that term as? It's not polypeptide, Okazaki, or polymer. The right answer is polysome. A polysome or a polyribosome is a complex of mRNA molecule and two or more ribosomes, which is formed during the active translation process. They were initially named as ergosomes in the year of 1963. However, who named it as polysome? It was Alex Rich who characterized and gave the name as polysome. Therefore, the right answer is option A. Next question here. Which one of the following is not applicable to RNA? Very easy question. We have four options here. This is applicable. This is applicable, this is applicable, this is applicable. Chargaff's rule is exclusively for DNA and not for RNA. It does not be applicable for, it's not applicable for RNA. So what does the Chargaff rule state? It states that the DNA from any cell of any organism should have 1:1 ratio. 1:1 ratio, that is the base, it's called the base pair rule of the pyrimidine and purine bases, and more specifically that the amount of the guanine should be equal to cytosine and the amount of adenine should be equal to thymine. So this is what the rule says. Therefore, it is not applicable for RNA. Therefore, the right answer is option C. And you have to your right side a diagram which clearly shows the base pairs that you can find in RNA. Adenine, guanine, cytosine, and thymine is replaced with uracil. That's the only difference between DNA and RNA. Next question here. Transformation was discovered by Dash, and this question is a direct question. I think many of you know what the answer is. Yes, the right answer is Griffith. Right? Griffith. He's called as Frederick Griffith, and that in the year of 1928. He's a British medical officer, and he described the process of bacterial transformation. He carried out an experiment with the help of Streptococcus pneumoniae, which is responsible for causing pneumonia in many living organisms, which includes the infection of the respiratory tract. So here, this organism was used for this process, and he carried out a method where he actually identified using two strains, that was R and S strain. The R strain got converted into S, so it's called the transformation process here. Therefore, who discovered this process is option C, Griffith. Next question. Uridine present only in RNA is Dash. Okay, any idea what it is? Uridine is a nucleoside that is present in RNA. Simple question. The combination of pentose sugar, nitrogenous base, and you have nucleoside. So that is what makes an RNA, and the same thing goes for DNA as well. So therefore, the right answer to this question is option B, nucleoside. Next question here. If one strand of DNA has a nitrogenous base sequence of ATCTG, what would be the complementary RNA strand sequence? Very easy question which was asked in the year 2012. So any idea what the answer would be? You have ATCTG. So we're asking the complementary part, right? Therefore, the right one will be option B. Sequence of DNA is this, then the sequence of mRNA would be UAGAC, as adenine pairs with uracil and guanine will pair with cytosine. Therefore, the right answer is option B. And I've just given you a DNA structure there which shows a double helical strand, and also you have the base pairs, nitrogenous bases, along with DNA. Next question here. The unequivocal proof of DNA as a genetic material came from the studies on Dash. So this was easy. This is an easy question because we already discussed about it in the initial part of this chapter. So they carried out an experiment with the help of a bacteriophage, where they saw that where an experiment by Hershey and Chase carried out with the help of bacteriophage, where they said that this genetic material is what giving the bacterium release the progeny. Therefore, we considered bacterial virus, which is also called as bacteriophage, and bacteriophage is the one which infects bacteria. They are viruses, but since they affect bacteria, it's simply called as bacteriophage. Therefore, the right answer is option D. Next question. Infectious proteins are present in Dash. So this is a direct question. Infectious proteins are rich in prions or rheons. These rheons are made up of proteins without nucleic acid, and that's why they are the casual agent of scrapic disease of sheep. So far we haven't identified any disease with humans or many other animals, but usually this is more associated with the scrapic disease of sheep, and here you don't have any specific nucleic acid or genetic material, it's just the proteins there. But these proteins seem to be infectious, and that is why they cause diseases or nuisance to the other living organisms. Therefore the right answer is option B. Next question here. T.O. Jenner discovered a dash. So the full form for T is Theodore O'Denner discovered the potato spindle tuber viriot, which is abbreviated as PSTVD. It actually comes like this. Okay, the first viroid ever identified was in the year 1971, and this spindle disease, if you see, it is a small circular RNA molecule, and Theodore discovered that the pathogen causing potato spindle tuber disease is not a virus, as it previously believed to be. It was more similar like just an RNA. Therefore it was free infectious RNA, which is option D, the right answer. Next question, which one of the following pairs of the nitrogenous base of the nucleic acid is wrongly matched with the category mentioned against it? And this question was asked in the year 2008. So if you clearly see here, you just analyze the options given here. Thymine, uracil, they are right. Uracil, cytosine, they belong to pyrimidines. Guanine and adenine, purines. Adenine and thymine is where they go wrong, because thymine belongs to pyrimidine and adenine belongs to purine. Therefore the one which is mismatched here is option D, which is wrong. Therefore the right answer is option D. With this we complete the molecular inheritance part 1. Hope to meet you in the next part with another set of questions and explanations. Thank you.